Guys, and welcome to Research Technique of the Week. This is your host, Miranda, and this week's research technique is RT-PCR. RT-PCR stands for Reverse Transcriptase Polymerase Chain Reactions. A little backstory of this research method is reverse transcriptase, which is an enzyme that can create DNA from RNA, was discovered in the 1970s. Then, in 1985, Carey Willis invented polymerase chain reactions. These two discoveries are what paved the way for RT-PCR. Now it is in use in all sorts of fields like biotechnology, cell biology, drug research, and many more. The purpose of this research technique is to create the complementary DNA sequence and then to make many more copies of it through PCR. A common use of this technique is to look at genes expressed in the desired DNA strand. Here's a quick synopsis of the process. First, the RNA of interest is isolated. Then, to convert this RNA to DNA, reverse transcriptase must react with it. This will form the complementary DNA strand. Then, a polymerase chain reaction must occur to produce many more copies. So first the DNA is split to create the single strands, and then the primers will associate with the strands, and the DNA polymerase will elongate them. The process is repeated, and the product is many more copies of the DNA of interest that now can be utilized. This is a diagram that better illustrates the process and its steps. There are endless amounts of experience and studies that have RT-PCR incorporated within them. Today, I will focus on two real-life applications of RT-PCR. One real-life application of RT-PCR is in cancer research. There was a study conducted in 1996 where RT-PCR was being tested whether or not it is a useful technique in being able to express and detect certain proteins that are correlated to tumors. It was concluded that RT-PCR was a sensitive method for detecting these cells and it causes less contamination between samples. The other real-life world application for RT-PCR is one that has become quite popular in the past 3 years, and this is the use of RT-PCR in the research and testing of COVID-19. An example of a study conducted is where scientists incorporated RT-PCR into an assay that tests for positive COVID samples and identifies what variant of COVID it is. I hope you enjoyed this week's segment of Research Technique of the Week. Thank you for tuning in and remember to tune in next week.